Bismillah Rahman Rahim. Another paper 3, March 23, paper 3 practical exam skills. Hasbunallah wa ni'mal wakeel. Allah alone is sufficient for us and He is the best disposer of affairs. Question 2, P1 is a slide of a train transfer section through a plant route. Draw a large plan drawing diagram of the region of the route on slide P1 indicated by the shaded area in figure 2.1. Use a sharp pencil. So this is figure 2.1. You're going to see a slide which has been sent by Cambridge and you will see a whole route in it, but you've got to just draw the quarter of it. Use one rule label line and label to identify the xylem. So I don't have the Cambridge slide. I don't know what was the Cambridge slide, but I have a rough idea. So I'm going to just give you a slide and I'm going to show you what was supposed to be done about this. Suppose the slide looked like this. Now, if you look at this slide, now we have to draw a quarter of it. So a quarter of it would be something like this. So a quarter of it, then you have to realize is you have got to do, there was these two lines here, then there is this line here, then there is another line here showing you, and then of course we can see these. But I mean, of course, you're not going to draw the xylem vessel, so you're not going to draw this. So then the plan of the tissue would be that you would draw a line here, and this would of course be the entire diagram would be this quarter portion of it, and you would have to draw it in a little more larger way because you'd have to draw it like this. For the 5 marks of this plan drawing, I've just kept the slide in front and shown it to you like this, but of course you'll have to look at the Cambridge slide. Now, what are the 5 marks for? The 5 marks are for uses most of the available space. So the space available to you, which is from the question up till the marks, this whole available space should have been used for you. So it should be a big diagram right from this point till where it's written 5 marks. So till this point, you had to remember this is that you have to draw a big diagram. So you'd have to do a diagram as big as from here till right here, and there's all this. So 5 marks. What are the 5 marks for? Uses most of the available space, no shading, draws correct section and no cells, draws minimum number of tissues, at least 3 layers of tissue, draws correct position of xylem, label line and label to xylem. So 5 marks and everybody should get these 5 marks. Then part 2 of the question, observe the cells in the center of the root on slide P1. Select a group of 4 adjacent cells. Each cell must touch at least 2 other cells. Make a large drawing of this group of 4 cells. Use one ruled label line and label to identify the cell wall of one cell. Now I've chosen this slide for a certain purpose because I want you to see that if we have to draw 2 cells from the cortex, then these would be the 5 cells that I would draw. Now, important thing which I want you to see is, you see this is one cell. Then there is this space in between. This is another cell. And then this is another cell. And we had to draw 4 cells. So you can draw this one. Now, in the question it said draw 4 cells, but you see this space in between which we have left. This is called the intercellular space because you see in the mark scheme, it says very clearly lines continuous, thin and sharp. Draws each cell touching at least 2 of the other cells. Draws each cell touching at least 2 of the other cells. You can see this one is touching this one and this one, at least 2. And then when you look at it, this one is touching this one and this one. And then if you look at this one is touching this one and this one. So at least 2 other cells. Then two lines drawn around each. I haven't done that as yet, but I just wanted you to see this is that you were supposed to leave this intercellular space here, which I'm just coloring it to make you all understand it. But otherwise, I don't want you to be showing this as a line, but I'm just telling you. So two lines around each cell and three lines where the cells touch. I'll just show you that. Detailed shapes of cells and intercellular spaces between cells. This is the intercellular space between the cells. And label line and label cell wall. So if you had to do this, then this would be the cell wall. Now this, of course, I'm doing it on a slide, but you actually have to draw these separately, and I'll just draw these for you and then have you look at it. This is the tracing of it. But then you should have drawn it much bigger than what I've drawn here. So it should be a big one showing these four cells, and that is how you should have drawn it. But the important thing is the double line and the three lines. So diagram something like this. Of course, you won't draw this one, but I'm just telling you how you would have drawn this and how you should have got this correct to get your 5 out of 5. There's 5 marks for this. Lines continuous, thin and sharp, draws each cell touching at least two of the other cells. Two lines drawn around each cell and three lines where cells touch. Detailed shapes of cells and intercellular space between cells, label line and label cell wall. Then coming to the B part of the question, figure 2.2 shows a photomicrograph of a transverse section through a root. This root is different to the root on slide P1. You are not expected to be familiar with this root section. Fine. Then it says identify 3 observable differences other than color between the root in figure 2.2, this one, and the root on slide P1. Record these 3 observable differences in an appropriate table. Now there's a very important rule that we follow, that the first column has to be features, then you can say slide P1, and then you can say figure 2.2. You can do it in any order, but the features must come first, and then P1, and then figure 2.2. Whether you write figure 2.2 before or P1, but it doesn't really matter. So you have to do the observable difference. Observable means you're not going to talk about something which you are imagining or which like something which you can't see, that is cylindrical or something like that. You can't see it's cylindrical. It's just a transverse section. So if you look at the features, now I'm going to bring in a picture of a slide which I think was the Cambridge. I said I think which was the Cambridge slide. So I don't know what was the Cambridge slide. I don't have an idea what it was. So I'm just sort of thinking that maybe it would have been this one. Let's say, let's think that the Cambridge slide P1 was something like this. So if you look at the first feature, which I'm going to talk about, first feature is position of the vascular tissue. Now in P1, the position of the vascular tissue is towards the periphery because you see it's more on the outside. And in this situation, it is in the center. So position of the vascular tissue towards the periphery in this situation and in the center in this situation. Then the size of the xylem vessels, these are smaller. You can see the xylem vessels are smaller, these are slightly bigger. Then the endodermis is not visible, it is visible in this one. It is visible in this one, it was not very visible in the Cambridge slide. Width of the cortex is narrower. This cortex is narrower than the cortex here. So look at the cortex here. The cortex here is this. As looking at the whole slide, I'm not saying just look at this one, but this one now occupies a very large part of the whole diameter of the root. Then epidermal layer was less intact. It's sort of a little broken, and this one is a more intact. So I'm just saying this could, I'm just giving you an example of it. I'm not saying this is the Cambridge slide, but what if this was the Cambridge slide? So I'm just teaching you how to do the comparison between the slide which came from Cambridge and this photomicrograph. So features, position of vascular bundle towards the periphery in the cord center, size of xylem vessel smaller, larger endodermis not visible, visible, width of cortex narrow, wide, epidermal layer less intact, more intact. So this gives you a way how we are going to compare. You can talk about the edges, you can talk about the center, you can talk about different width of the tissues, you can talk about is something visible or something not visible. So you've got to see more slides of stems and roots so that you can do a comparative analysis of them. And then coming to part 2 of the question, figure 2.3 is a photomicrograph of the same root section that is shown in figure 2.2. And then you can see the scale bar, which is 2 millimeter. Use the scale bar on figure 22.3. So this is the scale bar, please remember. To calculate the magnification of the photomicrograph and the actual diameter of the root section identified by the line XY. Show your working, use appropriate units. And this is for 5 marks. The magnification and the actual diameter. Now, very simple. I hope everybody knows how we do this. I'm sure you all know this. So if you measure the scale bar is 4 centimeter. This is 40 millimeter, but it's actually 2.0 millimeters written here. So 40 millimeter is equal to 2. So 40 divided by 2. So the magnification is 20 times. Then we had to calculate the actual diameter of the stamps because this is actually a total 5 marks question. So in the 5 marks, then we had to calculate. So what is XY? So XY is 11.3 centimeter, and then it will be 113 millimeter. Now you have to draw that, then you have to divide that by the magnification. So 113 divided by 20, because 20 is the magnification. So that will give you the answer, and you have to give that to me here in the actual diameter. So 113 divided by 20 gives you 5.65 millimeter, because you see I am giving you all the answers in millimeter. I am comfortable with this, but they have not specified any units, but usually we do give the answer in millimeter because you see the magnification is given in millimeter. So I take a clue from that this is what they're expecting me to give. But it in the mark scheme it just says units. So I mean, if you gave it to me in some other unit like centimeter, I would probably be fine with that as well. So this is how you would finish this question number 2, and this was for 19 marks, and this is a total 40 marks paper, and very easy to score full marks in the question 2. Maybe you can lose 1 mark or 2, but not more than that. So all the best, and thank you very much, and please do subscribe. I want to reach my 10,000 mark, and that is very nearby, so please everybody do subscribe, and thank you very much.